Hello, and welcome to the third lab talk in our four-part series to help you master cold storage. My name is Wilfredo Marin, Senior Product Applications Specialist focused on cryopreservation and biobanking. We've got some great resources for you today to download in one of the boxes on your screen, and I highly encourage everyone to explore the Discovery Expo, where you can complete your passport to science and qualify to receive a set of building blocks. Today, I'll be sharing some important and specific sample storage temperature information applicable to many research and clinical settings. So I'm drawing from my experience having spent several years in the laboratory performing this very same processing, and then asking how quickly and efficiently can these vital samples be stored long-term. So I'll start today with information about where we used to acquire these samples and reasons why our lab was best suited to process them in a standardized manner. And I can speak to this why it matters and what projects they would often support. Now, call out our top three sample types to better understand the storage temperatures as well as using our Thermo Fisher solutions. And lastly, I'll invite our special guest, Ela Summit, who's fresh off of announcing the Freezer Challenge Award winners for this year, and the My Green Lab Raising the Bar Summit in September. And then we'll both stick around for Q&A at the end. So off to a very quick start, I know there's a lot of bullet points on this slide, but the key takeaway is that there truly exists something of a three-step foundational approach to working with samples. Samples come from engagement with a specific community network, and these can be health systems in areas with high incidence of disease. And when they speak with their primary physician, they might encourage enrollment in a study with the overall goal of it being a diverse representation of the population. Next, samples then need to be processed. And labs and facilities can be optimized to work with very large volumes over extended periods of time. And then a typical scenario to establish a longitudinal cohort may be getting samples prior to diagnosis, then samples again during treatment, and then hopefully again after remission. So labs can store on site or share with other investigators to continue the discovery. And then lastly, on this slide, some of the analysis that's being done utilizing these samples includes genomic sequencing. It'll help find variants or mutations. There's predictive protein biomarkers of a health status at that moment in time, and or predictive treatment response to medications, which a lot of the larger biopharma companies can leverage to improve health. This next slide is a really quick schematic of how we used to collect samples in my previous role and facility. It really began with understanding who would be visiting the clinic for a routine appointment or had been admitted to the hospital for further testing or even in an emergency situation. So research samples were always second to the vials that were being obtained by the physicians. Those vials went towards the immediate processing to inform the course of treatment for that patient under the care of important health professionals. So meanwhile, our pediatric patients, for those research samples, they required a consenting process. And we would use broad language and ask a phlebotomist to obtain an additional 2 teaspoons of whole blood, potentially a pencil tip of muscle tissue or a sliver of skin from a biopsy. And then we would take that back to the lab for processing and store it where it could be analyzed and harness that collective sort of statistical power of having many, many samples. So they could be in a variety of tubes with colorful tops, like I'm showing here, but we always try to be consistent with the sample type and the volume. So our top three in my previous biobank facility were serum, plasma, and cells. And I mentioned that we could acquire tissues as well, and we had containers for saliva, urine, or even specific RNA stabilizers like Tempus tubes. But here, what I'm showing is that sera would come from a red top tube. We would obtain it and keep it at room temperature for a few minutes prior to centrifugation. And then when separated, we would aliquot it and make sure that there was no hemolysis or there were broken red blood cells. And these would be included for immunological or antibody studies where they would be stored long-term at -80 degrees. Next to those samples were also frozen plasma tubes or plasma samples. They would come from blue top tubes. They are spun almost immediately. In many cases, it would be a refrigerated type centrifuge, and we're trying to achieve the platelet-free suspension. This was like a clearish yellow. And then when we would use these for proteomic studies, the volumes were a lot smaller because a lot of those instruments could read samples and concentrations with lower limits of detection. So again, we only wanted to thaw out the amount that we would need for that particular experiment. And again, these would go side by side with the sample or with the serum vials in that -80 freezer. And lastly, on the far right here, we had peripheral blood mononuclear cells, or PBMCs. They came from yellow top tubes. They needed to be isolated, often in a biosafety hood as a Ficol gradient, and then we would count them. And we would aim to store cryo tanks after we had properly used a control rate freezer to bring them down 1 degree per minute to those cryogenic temps. And these type of cells could be stored for genomic studies. You want to start, you think PCRs or any type of sequencing, as well as propagating any future lines that would require the use of incubators. So these were our top 3, and of course they varied in the types of temperatures that they would be stored in. An additional example of optimal sample storage temperature is shown here for mRNA. This question was asked really early in the days of formulating the COVID vaccine with that novel technology and the stability of the different products that were available. And so I'm citing 2 sources below because we know how important it was that patients have confidence in the effectiveness of these vaccines. And so the papers here support the concept that all biospecimens to some capacity have degradative molecules. The activities of those molecules are strongly influenced by temperature, meaning that the storage conditions need to be selected so that those degradative molecules are inactive. So mRNA is highly susceptible to degradation with these RNases, and the paper is citing that the conformation of these vaccine components, such as the lipid nanoparticles, is more susceptible at warmer temperatures that are not -80. So if the key takeaway from this webinar is anything, it would be that optimal storage temperatures for biological cells are a function of the complexity of the sample as well as the biospecimen type. So we're able to support that there's a certain stability of mRNA. There's purity numbers that you can achieve with the different ratios, A260 or the 230. And then again, we support that by providing purpose-built units. So as I'm finishing up my section, I did want to add this somewhat strange screenshot of something I saw around 2 years ago. And it really emphasizes to me that despite everything we know, all the years of knowledge that gets passed down from researcher to researcher in the lab, there's still questions about how to store your samples, what temperatures to use. And it almost feels like a rumor mill of what's worked or failed and no one ever wants to try again. But as you'll see from our guest, and you'll learn from Ela later, there's a lot of new information and there's different types of interventions that can be implemented to improve the cold storage process. It is ultimately your choice, as well as the responsibility to uphold or modify that decision. But I hope you've heard here that there are plenty of people with expertise at Thermo Fisher to help answer those questions. And the equipment is built to serve and enable your cold storage needs. So out of all these little comments here, I kind of, my favorites are the last 2. You know, we have the rule, short-term storage is -20. If you plan on using it quickly, long-term will be -80. It's most important to avoid those freeze-thaw cycles. And the question has usually come up, well, where did -80 even arise from? Well, technically it did come from the way that people use dry ice to move their samples. And so about -78.5 or so is the sublimation temperature for dry ice, and that cold vapor right above it. So it kind of stuck. But as you can tell, there's still a lot of questions surrounding, well, what is the appropriate temperature? And if you maintain that temperature, are you able to achieve the same integrity with your samples? So finishing up here, again, being back in the lab, I outlined some really practical steps of how we better utilize our samples. We had everything. We have slides, skin blocks, the muscle I mentioned, sera, plasma, We had DNA, the isolated RNA, we had PBMCs. And overall, storing this variety of biospecimens also meant we had to know their optimal storage temperature requirements and much of what you heard here today to maintain their functional integrity. So in my early lab trainings, we always learned DNA was stable at -20. That's completely fine, and that's kind of what we stuck with. There's also antibodies and kits that can't be frozen, so they have to be at refrigerated temperatures. And I showed an example of mRNA needing to be at -80. So sometimes there's media or chemical reagents that are expired. It's very easy to get rid of those and not have them get in the way of the items that we're using more on a regular basis for that sample processing. So much of this took a little larger effort, and we have to rally the team to organize, and so we could execute on our experimental goals. So ultimately, if you can educate and engage your staff, it can lead to both increased productivity in the lab as well as job satisfaction. So with that, I would really like to, I'm very happy to introduce Ela Summit, who is the Sustainability Programs Manager for MyGreenLab.
Hi, everyone. I'm Ela from MyGreenLab. Pleasure to be here today and to speak with you. I'm going to talk a little bit about MyGreenLab and what we do and how we support cold storage, specimen, and sustainability. I'm covering a few of the topics that Will went over in a little bit more detail. I hope you find this useful. So at MyGreenLab, we're a nonprofit organization started about more than 10 years ago, and we are dedicated to making the world's lab more sustainable and having less of that environmental footprint while maintaining scientific integrity in all the work that we do in the lab. We have a vision to create a world where science is conducted in a way where we're not only benefiting the health of the people, but of the planet as well. So one of our programs is the Freezer Challenge. We are really trying to get people engaged. It's a free program. It is a flexible program, voluntary, trying to get people in the lab a little bit more engaged with maintenance of freezers, inventory management, and really implementing those sustainable practices in their daily work week. A lot of times cleaning up freezers is not the #1 job that anybody enjoys to do, but it is absolutely critical. So why do we focus on cold storage? Well, it's been shown really that second only to fume hoods, this equipment category is the largest plug load in most lab buildings. So one ultra low freezer is about 10 to 30 kilowatt hours per day, and that's about as much as it takes to power one US home for a day. So a lot of energy, and if you multiply that with how many typical freezers we see at -20 or -80 in one lab alone, as you probably already have seen, this is multiplied quite to a large degree. This is just a quick glimpse at how our Freezer Challenge has been going really in the last year. We have been able to get more than 35 countries engaged, more than 240 participating organizations from around the world, which is really exciting to see. It's not just a US, Canada, sort of Western world effort. Places everywhere are really wanting to get involved and figure out how they can lower their electric bills, be more efficient in the lab. And with that in mind, last year, we are now coming to a total here of 31.8 million kilowatts an hour of energy saved. So just putting that in perspective, that is the same as 22,000 metric tons of CO2 equivalent. And we've been doing this competition since 2017. So the energy savings just keeps compounding. So if you want any more information on how to sign up for this, of course, please feel free to visit us, and I can certainly help you out with that. But going just a little bit into the actual cold storage practices, we really want you to be able to picture yourself how you can get involved and what types of activities we're really looking to doing in the lab. So some of these activities are more directly related to energy savings. Some are a little bit less. But all in all, they're really to make your processes more efficient without losing any of that sample integrity. So powering down, defrosting freezers that you don't need, the general maintenance of cleaning out, defrosting, those are sometimes done by facilities, those are sometimes done by lab people, but definitely something you want to keep doing on a regular cadence. Converting into high density storage boxes, and really making sure they're efficiently packed in that box. We don't want samples spread out. Sometimes better barcoding, maybe quicker barcoding if it's something where you know where you're going in the freezer, you know which samples you're looking for, you don't have to have the door open as long. All of these things can really help keep the temperature stable in your freezer, and keep it working for long term as well. As Will alluded to, temperature tuning is one of them that we definitely focus on in our freezer challenge. Again, because we see that change from -80 to -70, showing quite a huge energy savings. This varies now depending on freezers as they're becoming much more efficient, which is absolutely wonderful. But again, it really depends on your sample type. Not all samples need to be kept at -80. Some are absolutely fine being kept at -70. And you can have different freezers at different temperatures if that makes it easier. So we actually have a database at our website, on our site, which for some resources here that really can help you decide where you want to put your samples. So some of our databases are collected by just our users, people around the world who want to contribute, and they indicate what type of samples they've been seeing and for how long, at what temperature they seem to be holding up at. And some of it is dependent, of course, on downstream testing, depending on what you're looking at. Some temperatures might be better than others. So again, that tends to be something that's a little bit harder to get all of your freezers, at -70. So certainly it's not meant to be all freezers. Start with one freezer, see how it goes. If there's any sort of issues or worry about what could happen to the samples themselves, start with something that isn't as in dire need, then that would certainly be the way to go. But we do tend to sort of look at your freezer temperatures and see what you can do with them. They're not always as set in stone as you might believe. And of course, having a backup plan, no matter what temperature you're keeping your samples at, having an extra freezer, having a plan if something goes wrong in the lab, freezers go down, we definitely want to make sure that you have somewhere where you can move your samples or some sort of backup power as well. So that's really it for me. I'm going to pass it back to Will.
Thank you so much. Description of the work that My Green Labs is doing, as well as sharing some of those resources. I was really impressed by that database you mentioned. It was one of the first things that I looked into because it was compiled from lab managers all over the world, and just an entire list of samples that had additional information about those temperatures. Is that something? I guess we're ready for questions Q&A, but maybe before we get started, is that something you could elaborate on, and how people have used it, or what's been most challenging about getting that out there, and talking about freezers is not just -80s, but maybe ULTs, any thoughts on that?
Yeah, one thing that's definitely been um, challenging, I would say, with having that database is maintaining it and how often um, the samples are being tested for viability after they've been put in a different temperature. So we do need processes for that, and that's going to again depend on downstream testing. Um, if maybe after a year, 6 months, rechecking those samples, see if they're still viable. And a lot of times in research labs, we don't always need to do that, but in more regulated settings, that is something that's a little bit more important to keep looking at, and something that you need records of as well. You will be audited for. But Yeah, I would definitely encourage anybody on the call to add to that database, look at it, check it out. There's papers as well, indicating what type of storage samples are going to work better. Sorry, temperatures are going to work better and which won't. So,
I agree. I think people tend to recommend pieces of equipment or instruments that they like and they understand. They also want to hear from, very knowledgeable scientists about the proper temperature. And we can say it's a blanket statement. It's always minus 80, but again, you said it varies, and even for those three types of blood vials that we would collect, each one had its own process. Each one had a temperature depending on what was being stored. So no, great. That's terrific. And I really like that you started to hint at best practices, because that will be our last lab talk of the series. You almost gave a not a sneak peek, but almost like a cheat card of how people have won the freezer challenge, doing some of those, taking some of those actions. Do you find that any one of those has had more success in really driving the efficiency of a lab storage of your little cheat sheet? Yeah, it's on
yes.
the shown on the screen now. Thanks.
Yeah, so there's definitely some actions that are going to equate more to energy savings, some a little bit less and more in terms of efficiencies and timing, just how long it takes to retrieve samples. I think that's where barcoding and new inventory methods really come in handy. So we've seen a lot of labs completely change their systems, consolidate samples from 5 freezers into 1 just by cleaning. So regular cleanings, getting rid of those old samples and actually having like a practice in place. We go back in and go through our samples every, biannually, something like that, so that there's actually something that people can stick to. This is how many samples we got rid of, and that's part of our freezer challenge too, that we have those numbers that you can kind of record and see, like, how many samples are we really throwing out here? And usually it's boxes and boxes. I think that would be the probably one of the number one things we've seen as people leave the labs. Usually they leave their stuff behind as well. So just even sort of like an offboarding process there of somebody who works in the lab, like, where's all your stuff? Can we get rid of some of it? And who's in charge of those things?
I agree. It's a touchy subject because we've been taught, at least I was always taught, everything you collect is important, right? It's a very precious type of sample that you can never get again, but you have to do something with it. And if you can use it rather than throw it away, that's obviously the preference. But at some point, yeah, that's a difficult decision. You want to use it rather than lose it. That's always one of the impressions I heard. And then if you put garbage in your facility, like improperly processed, you're going to get garbage data out of all that information. And so I have one last question for you, and feel free for the audience to type anything in the conversation. We spoke about this just before we started, in terms of, we have specialists and representatives from Thermo Fisher joining. We have customers all over the world. Um, is there something that they can do with My Green Lab? We talked about a little bit about certifications if they have lab, or the is it a certificate or some type of action that they can do to participate in some of the initiatives that you have, because they don't have freezers of their own, but they want to learn ways to be more sustainable in their everyday lives. So
Mhm. Absolutely. Yeah, just to even start, of course, with the freezer challenge, we've had questions come up, "I only have 1 or 2 freezers in the lab. Is it really gonna be that useful?" Yes, it can. There's those single actions do add up. I think if you only have 1 or 2 freezers, it might be even argued the opposite, that it's even more important to take care of those freezers. If one goes down, you certainly need a backup. So, in terms of the freezer challenge, absolutely free program, easy to sign up. If you're in the lab and you're somebody who really wants to take charge and kind of start looking more at sustainability, We do have an ambassador course, which is free as well for everybody to really get educated on quite a few topics. We cover cold storage, of course, and we cover a lot of equipment, energy, water savings, things like that. Plastics are a huge one. Of course, we all know that if we've ever stepped foot in a lab, a ton of waste there. So, and that course is designed to, it's about, I would say it takes about maybe a full day to get through the whole thing. But you get more educated on simple tips, communication, how to talk to your peers, colleagues about sustainability, and feel confident in what you're saying as well. It's not just, you know, it's not just words. These actions really lead to savings, and that's really important, of course, to our companies and overall efficiency. So, yeah, happy to share any information with that you would want, and hop on our website, and happy to help with that. Yep.
Well, great. Thank you. It's been a pleasure speaking with you, and I look forward to hearing more from My Green Labs. And obviously, I hope everyone listening is very motivated to join this freezer challenge for the upcoming year and reach out to Thermo Fisher representatives to see how we can support any of those, temperature requirements as well as long-term storage. So everyone very much for attending, and make sure to join our last lab talk where we will talk about walking through best practices for cold storage equipment. Thank you, everyone.